Hello students, today we'll be learning about pentose phosphate pathway. I am Shaista from Department of Studies in Microbiology, KSOU. Uh, to have a brief idea about pentose phosphate pathway, we must know about glycolysis, which we had dealt in earlier in the classes. Please go through it, have a rough idea, and then we will proceed with this pentose phosphate pathway. Okay, let's begin. Pentose phosphate pathway is also known as PPP pathway in general. It can also be called as hexose monophosphate shunt. Okay, where does this occur? It occurs mostly in all cell types and tissues except muscles. Muscles do not use this pathway. Majorly, it occurs in liver, where 30% of the glucose in liver is metabolized by this pathway. And it even occurs in erythrocytes, that is our RBCs, red blood cells. Okay, it occurs majorly in cytoplasm of the cells. Okay, so what does it produce? It produces the reduced form of NADPH. NADPH is very essential in various pathways like fatty acids, where 50% of NADPH produced by pentose phosphate pathway is utilized for fatty acid synthesis. It is also utilized in oxidative stress hemostasis, majorly in liver and RBC to protect them from the oxidative stress, and it is used in the generation of cytochromes P450 enzymes. Okay, so where NADPH is used is highlighted over here. This pathway is also producing pentose sugars, triose and hexose sugars. All these, especially pentose, is used for nucleotide synthesis. So this is the importance of this pathway where its products are being used in various metabolic synthesis. Okay. So now, students, let's understand that pentose phosphate pathway is an alternative pathway. Generally, we know that in glycolysis, glucose is converted into glucose 6-phosphate. This is the first step of glycolysis. With the enzyme hexokinase, one ATP molecule is used over here to phosphorylate glucose into glucose 6-phosphate, so that glucose 6-phosphate is captured within the cell and it does not leave the cell easily. And once glucose 6-phosphate is captured within the cell, it can either undergo general process of glycolysis where glucose is converted into two molecules of pyruvate, and then the citric acid cycle, and then the electron transport chain. All these are interlinked. But depending on the need of the cell, there is an alternative pathway, that is pentose phosphate pathway, because of the various biochemical synthesis we need, like purine, pyrimidine, or the fatty acid synthesis, or various other cell needs. This alternative pathway is adopted, where this same glucose 6 phosphate is used as substrate. I hope you understood. So students, now let's discuss the important points of pentose phosphate pathway. This is also called as phosphoglutamate pathway and hexomonophosphate shunt. It always runs parallel to the glycolysis. It generates NADPH and pentose, that is nothing but 5 carbon sugars, like ribose 5-phosphate, which is a precursor of the biosynthesis of nucleotides. It involves the oxidation of glucose, and mainly it is anabolic rather than catabolic, whereas glycolysis was catabolic and pentose phosphate pathway is anabolic. The pathway is very important in red blood cells as well as liver. And this pathway is divided into two important phases. The first phase is called as an oxidative phase, in which NADPH is generated, and the second phase is called as non-oxidative synthesis of 5-carbon sugar. So generally this cycle is divided into two phases. One is the oxidative phase and the another one is non-oxidative phase. Now let's study each phase in detail. So the first phase is oxidation phase, where glucose 6-phosphate undergoes dehydrogenation and decarboxylation to yield pentose, that is our ribulose 5-phosphate. In first phase, glucose 6-phosphate is getting converted by various steps into ribulose 5-phosphate. And this ribulose 5-phosphate is precursor for many other biosynthesis process. This we have to understand. Our aim was to generate ribulose 5-phosphate, which is an important precursor. And this is generated from glucose 6-phosphate in the first phase of pentose phosphate pathway in the oxidation phase. Now coming to the second phase. In second phase, ribulose 5-phosphate is converted back to glucose 6-phosphate through various series of reaction, and it can thus again go back and follow glycolysis. Okay, and this is done by importantly by two enzymes, that is transketolase and transaldolase. You have to remember these names, like transketolase enzyme and transaldolase enzyme, which we will be talking in detail in coming class. Okay, hope you're clear with the difference between the two phases, the oxidative phase and the non-oxidative phase of the pentose phosphate pathway. Students, I'm trying to give you a very simplified version of this pathway. If you go through your notes or some other diagrams, you find it very complicated to understand. So I have generalized the stuff for you so that you can understand it in a very easy way. Okay, so let's proceed with this pathway. The first step after the glucose 6 phosphate is formed, the first step in this pathway is that the glucose 6 phosphate is getting converted into glucano 1,5-lactone 6 phosphate, and it is done by glucose 6 phosphate dehydrogenase. Same glucose 6 phosphate dehydrogenase, where NADP is reduced to NADPH. There is a generation of NADPH. In the first step, here in this cycle, NADPH is generated. Now coming to the second step in the cycle, where glucano 1,5-lactone 6-phosphate is converted into the 6-phosphogluconate. Okay, and this was our second step. The third step of this is ribulose 5-phosphate is generated, which is our important precursor. It is generated due to the action of enzyme 6-phosphogluconate dehydrogenase. Here also NADP is reduced to NADPH. So our two precursors, that is NADPH and NADPH, these are used as precursors in various biochemical synthesis. In the first step they are generated, and again they are generated in the third step of the cycle, where ribulose 5-phosphate is generated, which is an important precursor for other biochemical synthesis pathway. Now this entire phase is called as an oxidative phase of the pentose phosphate pathway. Hope you are clear and you understood. There are three steps in this, and the first step is generating NADPH, and the second step is also the third step is also generating NADPH, where we got our important molecule, that pentose, that is ribulose 5-phosphate. Now this ribulose 5-phosphate can be used in various biochemical biosynthesis cycles. Okay, now let's discuss one of the possibilities over here, and from this starts the non-oxidative phase. Okay, after ribulose 5-phosphate is generated, then starts the non-oxidative phase of the pentose phosphate pathway cycle. Now coming to the second phase, is that ribulose 5-phosphate is converted into xylose 6-phosphate by the action of enzymes ribulose phosphate 3 epimerase. Okay, and this here various steps of reaction is taking place where ribulose 6-phosphate is converted into fructose 6-phosphate. Various steps are taking place, and here various enzymes are acting, like our transketolase and transaldolase are acting over here so that they convert xylose 6-phosphate into fructose 6-phosphate. Now, where have you studied fructose 6-phosphate? Try to remember it. Yes, we have studied fructose 6-phosphate in glycolysis, where it is involved in the third step of our glycolysis cycle. Okay, and from here, as said, our glycolysis is proceeding ahead. So the conversion of xylose 6-phosphate into fructose 6-phosphate, and from there our continuation of glycolysis. This is our one of the possibilities where xylose 5-phosphate can be used. Now let's look at the second possibility of where this xylose 5-phosphate can be utilized importantly. So our second possibility is conversion of ribulose 5-phosphate in ribose 5-phosphate by the action of enzyme ribose phosphate isomerase. Okay. After this conversion, ribose 5-phosphate is converted into 5-phosphoribosylribosyl pyrophosphate. This is called as, this is further used into the synthesis of pyrene and purine synthesis. So this is an important step where this precursor, ribose 5-phosphate, is used for the synthesis of purine or the pyrimidine synthesis. Now, one possibility was that it was converted into xylose 6-phosphate and from there fructose 6-phosphate because of the action of the enzymes transketolase and transaldolase. This is possible where fructose 6-phosphate is generated. Here, various steps are involved over here. And this will be discussing next in detail. What are these steps existing over here due to the action of transketolase and transaldolase, where xylose 6-phosphate is converted into fructose 6-phosphate. So this is a simplified version of pentose phosphate pathway where you have various deviation. One is the ribose 5-phosphate is going for ahead with glycolysis, and it is also going ahead with the pyrene and the purine synthesis. Okay, I hope it is clear for you to understand pentose phosphate pathway. This diagram can be used to explain uh, pentose phosphate pathway for examination as well, a generalized diagram. So you can use this in answers. You don't find this in your notes, but you can use this diagram to write in your answers in exams. Okay. Now let me summarize to you, student, what I had explained you earlier. Here in the diagram, you can see that glucose 6-phosphate is our precursor, which is resulting in formation of ribulose 5-phosphate, where two molecules, that is one in our step 1 and one in our third step, are used. Therefore, 2 NAD plus is converted into 2 NADPH. And from here, ribulose 5-phosphate is getting converted into fructose 6-phosphate. Okay. This is our second phase, and this is our first phase. And in between, you have series of steps, which I told you earlier. These series of steps are because of the catalyzation reaction of the enzyme transketolase and transaldolase. Okay, and these are the steps where ribose 5-phosphate, xylose 5-phosphate, and glyceraldehyde phosphate, and streptoheptulose phosphate, fructose phosphate, and erythrose phosphate are all generated. These are our intermediate steps. I hope you are getting clear. So all these steps are involved. We will see this now in detail in the next slide. Okay. what is happening, we'll see here. So now coming to the oxidative phase, the oxidative phase is a non-reversible phase. It generates NADPH. We know that in step 3 and step 1 of oxidative steps, these are generated. Here, the glucose 6 phosphate undergoes hydrogenation and decarboxylation to give a pentose, that is ribulose 5 phosphate, which is converted into ribose 5 phosphate. This ribose 5 phosphate is used in various synthesis of pyruine and pyrimidine. That is also explained in the earlier slide. Okay, now the overall equation you can see is that glucose 6 phosphate plus 2 NADPH, one is used in step 1 and one is used in step 3, plus water gives rise to ribulose 5 phosphate, carbon dioxide, and 2 NADPH, which is our important precursor generated over here for other cycles, and hydrogen. I hope you're clear with this equation because of my earlier explanation. Okay. Now this is our oxidative phase, and in non-oxidative phase, this ribose 5 phosphate is converted into glucose 6 phosphate. I had told that two main enzyme acts in this. That is trans-ketolase. You can tell TK, and this is trans-aldolase, TA. Okay, this trans-ketolase transfers two carbon fragment. Okay, and trans-aldolase transfers the 3 carbon fragment. Now, if I have to tell you, you can see over here, to begin with, this ribulose 5-phosphate is converted into ribose 5-phosphate, and it is also converted into xylose 5-phosphate. Both are C5 carbon compounds. You can see here C5 and C5. Now what happens here? A C2 fragment is removed from one of these and attached to the one of these. So you can take a C2 fragment is removed by the action of enzyme trans-ketolase. It is removing one of the C2 and attaching here. So this becomes a C7 carbon compound. And when C2 is removed from here, it becomes C3 carbon compound. That is, C3 carbon compound is nothing but our glyceraldehyde phosphate gap, called as gap, which is nothing but an intermediate of the glycolysis. I hope you have seen this in glycolysis, right? In our step 5 or step 6, we have seen this. So this is a C3 carbon compound, and here it is a C7 carbon compound. How is this happening? This is happening because a C2 fragment is removed from here and attached to this by the action of the enzyme trans-ketolase. Now we have got a C7 carbon compound and the C3 carbon compound. Now what happens in this is that this intermediate is getting converted into both the fructose and erythrose. And how is this happening? This is happening when a C3 carbon fragment is removed. And when the C3 carbon fragment is removed by an enzyme, trans-aldolase. Trans-aldolase is acting here. It is removing C3 carbon fragment from the C7. So it is attaching to this C3 and it is becoming fructose C6. So 6 carbon compound. C3 plus 3 is 6 carbon compound. Now C7, 3 fragments are lost. C7 minus 3 is C4 carbon compound. Okay, so we have got a C6 carbon compound and a C4 carbon compound. And this C6, that is our phosphorus 6 phosphate, is following the glycolysis. It will go into glycolysis. So it is cancel over here, it will enter glycolysis. Now what about this C4? This C4 with again our C5. Okay, that is our xylose 5-phosphate. This one itself, this xylose 5-phosphate. Same thing over here. This C4 and C5, again here C2 fragment is removed. A 2 carbon fragment is removed by the action of enzyme trans-ketolase. So it is removing from this C5, two fragments are removed. So it becomes C3 carbon compound, that is nothing but our gap, glyceraldehyde phosphate. And this 2 from here is going and adding over to this C4 carbon compound over here. And again, it is becoming fructose 6 phosphate, which is a C6 carbon compound. Again, this compound is going and following the glycolysis cycle. Okay. So this is also entering the glycolysis cycle. This is also entering the glycolysis cycle. And we know that glyceraldehyde phosphate is also entering the glycolysis cycle. So, and from this, the glycolysis will continue. I hope this diagram was clear for you to understand how the transketolase and transaldolase are acting by removing a two fragment from every time. First, there was a removal of two carbon fragment by transketolase, and then it was three carbon compound removal from the compounds by the action of transaldolase, and again a two fragment compound removal because of the transketolase. There are three reactions, three steps. First was by transketolase, then transaldolase, then transketolase, and then we got our fructose 6 phosphate and gap. Okay, our end products were C6 compounds and C3 compounds because of the action of our transketolase and transaldolase. I hope students you are clear with this diagram and you have understood the series of conversion steps where ribulose 5-phosphate is converted into fructose 6-phosphate because of the action of the transketolase enzyme and the transaldolase enzyme. Okay. Now, students, a generalized summary of what I had explained earlier, the three series steps which are involved in the conversion of our ribose 5-phosphate into fructose 6-phosphate. These are the generalized steps. You can see this 5 carbon compound and the 5 carbon compound, which is nothing but our xylose and ribulose 5-phosphate is getting converted into 3 carbon compound and a 7 carbon compound, where our enzyme transketolase is acting and it is removing one of the 2 carbon compounds from here and attaching it to the 5 carbon compound. Therefore, 5 minus 2 is 3, 5 plus 2 is 7. Okay, this is because of our action of our enzyme transketolase, where this is glyceraldehyde 3-phosphate and this is pseudoheptulase. Okay, now again taking this 2 in the our next step, that is our 7 carbon compound and the 3 carbon compound. Now our enzyme transaldolase will act. What does transaldolase do? It will remove a 3 fragment fragment and attach it to the other. So from 7, it is removing a 3 fragment and it is attaching to a 3 compound carbon. Therefore, 7 minus 3 is 4 carbon compound. 7 plus 3 is 6 carbon compound. Now, this is 6 carbon compound is nothing but our fructose, which will go off into the glycolysis. Okay, it is entering into the glycolysis from here. Now, our remaining is our 4 carbon compound, that is nothing but our erythrose. Now this 4 carbon compound erythrose and our 5 carbon compound, that is our first one, the xylulose 5 phosphate. These two are taken together because this 6 fructose 6 phosphate has already entered the glycolysis. Okay. Now in these two, again the enzyme ketolase is acting. So it is transferring the 2C fragment, that is 2 chain carbon fragment from this 5 to the 4 carbon compound. So 5 minus 2 is nothing but our 3 carbon compound, that is our glyceraldehyde 3-phosphate, and 4 plus 2 is 6 carbon compound, that is nothing but our fructose 6-phosphate. This fructose 6-phosphate enters our glycolysis and this glyceraldehyde 3-phosphate also enters the glycolysis. Therefore, on cancellation, 3 cancel 3, 7 cancel 7, 4 cancel 4. So, 1, 2, 3. That is 5 carbon compounds are resulting into 2 6 carbon compound and 1 three carbon compound. Okay, this is nothing but fructose, this is nothing but glyceraldehyde 3 phosphate, and these are our pentose. Five carbon sugars are called as pentose. Okay, this is an overall explanation of pentose phosphate pathway. I hope you are clear. I have tried to give you a very simplified version of the cycle, explained you it in detail of various steps which are involving in this. I hope students you have understood. To understand this, you should have prior knowledge of glycolysis. Once you understood glycolysis, it is very easy for you to understand this pathway as well. So I have completed glycolysis. Now I have completed pentose phosphate pathway, and I will start taking other pathways which will be helpful for you to understand various steps. Okay, various other cycles. Okay. Thank you, students, for patiently listening to my class. I hope I have done justice with this cycle called pentose phosphate pathway. I have tried to simplify it. I hope you understood this. I hope to see you again with various cycle with easy explanation. Thank you, Anand.